Call me Larry. I prefer that to Lawrence. I'm living proof that you can't be too old to go back to school. I'm going to be talking about some new approaches that I've been working on for studying natural enemies of mountain pine beetles in standing trees. And let's see how this works. Yay! Okay, so why do we want to study them in standing trees? Well, thank you, Groucho Marx. Bark beetles and their natural enemies spend most of their life cycle under the bark of trees. A mountain pine beetle is out in the open environment for about 3 days out of its 1-year life cycle. Most of the action is happening in standing trees, and it's a very difficult system to study. So we don't have adequate methods for really preserving the nature of this system. So I'm looking at a couple of different methods that can be used that would be useful for learning some things about mountain pine beetle that we don't have a really good handle on. One of them is predator exclusion. It's never been used with mountain pine beetle. And I'm going to explain this more fully later. And the other one is using emergence traps, putting emergence traps on the standing trees and seeing when these other insect associates and what they are and when they emerge from the trees. Not been done with mountain pine beetle. So, a little bit about the role of the bark beetle natural enemies. Let's look at these epidemic bark beetles have 2 different population phases, and it's really a total complete minimum number of or percentage of bark beetles that actually do this. So there's like over 5,000 known species of bark beetles in the world. There's maybe 3 dozen, 4 dozen worldwide that actually kill living trees and cause epidemics. These types of bark beetles have 2 different population phases. They have a non-epidemic phase and an epidemic phase. The non-epidemic phase, the insects are not present in large enough numbers to overcome healthy trees. And that's really important that their numbers can increase to where they pass a tipping point where there are large enough numbers of bark beetles in the environment to overcome healthy trees. And that's really important for this epidemic phase because they reproduce a lot faster when they're attacking healthy trees. It's a much better resource for them. So then the epidemics will fade off. You get another tipping point. Populations get too low to kill healthy trees. You're back to this non-epidemic state. So what the natural enemies do is that they maintain these low populations. They're one of the main factors in maintaining populations low enough so that these bark beetles don't get out and start killing healthy trees. But for a number of different reasons, there's lots of different factors involved. Sometimes bark beetles pass a tipping point and we get enough bark beetles in the environment to start killing healthy trees. And so these natural enemies kind of determine where this tipping point is. They make it more difficult for bark beetles to achieve those kind of populations. Once we're in the epidemic phase, now this is where the $1 million question rests. In the epidemic phase, they definitely reduce the rate at which these bark beetle populations increase. But we don't know by how much. Pretty much in the system with mountain pine beetles, back in the last century, the 20th century, my God, that was a long time ago, wasn't it? In the 20th century, mid-20th century to about 1980, through that period, people started to conclude that, okay, these natural enemies don't make any difference during an epidemic. Today, more and more people who are working in the field are beginning to believe that, yeah, they do have an effect on the severity of epidemics. And the $1 million question is, how much is that effect? And so some of these methods that I'm going to talk about, particularly the predator exclusion experiment, is designed to measure that. When we go back past this other tipping point, Well, there's another tipping point. There's another thing that can happen. We get back, the populations collapse, they get too low to kill healthy trees. Well, they can rebound. And now we're in a sad state again. But natural enemies also prevent that from happening. So there's another, these are the roles of natural enemies. So both the methods that I'm working with depend on putting enclosures on standing trees. The predator exclusion experiment and the emergence traps have both been used in other bark beetle systems, and this particular enclosure is an emergence trap. It's on a tree. It's made with opaque cloth, and both types of enclosures use this kind of material. Opaque cloth being a weed cloth or shade cloth. Inside that enclosure, it's dark. These are collecting jars. They're clear, and that's the only source of light to insects that emerge out into that enclosure. And so insects are attracted to light. That's the way this emergence trap works. It traps the insects. They go to that light and get trapped in those collecting jars. Well, when I was first starting the study 3 years ago, I looked at these designs and I thought, well, there's something wrong with this. This is disturbing. This is upsetting the natural habitat. And this blocks light and air. It's altered the thermal environment, and insects being dependent on their environment for their body temperature. It affects their rates of development, and it affects how they relate to each other. And there's a whole community of insects in here. So when I first started this, making these emergence traps, I made them with a material that addressed this problem. But there's another problem that I didn't anticipate, and that's that, well, bark beetles, you know, when they land on a tree, where do they start their tunnel? They go into crevices in the bark, and that's what stimulates that burrowing behavior. Well, this type of an enclosure has lots of creases and folds and tight spots in it, and encourages burrowing behavior. And if a bark beetle can chew through wood, it can certainly chew through any kind of cloth. So this is another failure of this type of design or material. Well, my first emergence traps were a modification of this design. The very first ones was an enclosure around the entire trunk of the tree, just like the original one that I sort of used as my model. It was made from no-seam cloth. You can see it's practically transparent. Light gets through there, air gets through there. Well, that gives me another problem. How do I collect the insects now? I can't use light to collect the insects, to attract them to the collecting heads. But I used another factor of insect biology. Some insects, when they're in an enclosure, like to fly to the highest spot. Some of them like to go to the lowest spot. So I put my collecting heads high spot and low spot. And this worked pretty well. But all along here, you can't see it, but there are pleats that we had to put into this to make it fit the tree, because the tree's narrower here than it is here. Every one of those kind of areas, if it was a tight spot that beetles could get into, they started to chew. Found holes, found lots of holes here, found lots of holes here, found lots of holes along this sewn seam, found lots of holes along that. So the next design that I tried, still using the same material, eliminated those kinds of tight spots. I never deployed this one because that year, first year, I was doing predator exclusion experiments and I was too busy to bother with that. And besides, this suffers from another problem that this one also suffers from. Both of these have to be custom fit to the trees. So tree of a different size, you have to be custom fit in the field. A lot of work, a lot of time out in the field that could be better spent doing something else. So the last design, the successful design, this one right here, this is an enclosure that gets stapled or screwed. I originally used staples, now I'm using screws. Is screwed to the tree. The enclosure is made out of screen. This is the original design for mountain pine beetle or other bark beetle emergence traps. These are emergence traps that were designed to capture bark beetles as they emerge from the trees, so they could measure how many beetles, what was the density of beetles under the bark, and when did they emerge. And it was made with window screen, but doesn't work for the other associated insects, because many of the associated insects are much smaller than the bark beetles, and they go right through the screen. So I used a much, much finer screen. Insects emerge, and they fall down into this collecting jar at the bottom. Works really well, very cool. And it still preserves, some light still gets there, and air gets to the, I think this still alters the thermal environment, but not nearly as much as that black shade cloth. Okay, so that's my working design. Now for the predator exclusion. Well, predator exclusion has been used with several bark beetles in the past. It's been done with dark enclosures like the ones I showed you, cloth enclosures. It has demonstrated a really large effect on the survival of the beetles. So if you exclude predators from the beetles, you get a much higher survival. So I thought, well, this is going to work good for mountain pine beetles. I'll try it on them. So here's how you set one of these experiments up. You start with putting an exclusion enclosure. So from the bottom of this black cloth to the top of that black cloth at the top, no insects can reach the trunk of the tree. And then you put a lure, a bark beetle lure on the tree, because now you've done all this work and you want your tree to be attacked, right? You don't want that work to go to waste. So you put a lure on there, and when the bark beetles start to attack the tree, there's an opening in this enclosure. It has a sewn seam. You open that up, and you trap live beetles, you put them in that enclosure, sew it back up, and now you've got a part of the tree where bark beetles are living but are not exposed to predators. And then the following year, prior to beetle emergence, and here beetles emerge, the mountain pine beetle emerges late July, early August is the time of the heaviest emergence. So you want to cut this tree down before that happens. And then you're going to cut the tree into sections. So here's the section with the exclusion enclosure. That section contains beetles that grew up without predators. And then you're going to take a section from here. Those are beetles that grew up with predators. And then you're going to put those into rearing cages where you rear the beetles out and you count beetles, you compare the number of beetles, and you've got a direct measure of the effect of predators on the survival of mountain pine beetle. Well, I was really proud of these cages, these rearing cages. Because, man, I could put one of those together, $15 worth of materials. Really cheap to build. But they suffered from the same problem as those cloth enclosures that were on trees. Bark beetles chewed their way out. Bark beetles didn't realize that they were supposed to all go to the light here. Some of them just chewed their way out of the box. So went to the brass mesh material again for this. It's a lot more expensive. It's a little bit more difficult, but now here's the rearing cage. And I've got some bolts of wood, pieces of wood in some of these out here in a barn on the beef farm. And the beetles have been emerging. They fall to the collecting jar. In fact, all the insects that are in that piece of wood fall to that collecting jar. So not only can I get measurements of numbers of beetles, but I can also see what's associated with the beetles. It's really pretty cool. This is still going. I have the results from last year's experiment. This year's experiment, I don't have the results yet, but this is what you all want to see, right? Okay, this is the exciting part. It did, even with all the imperfections in the system last time, it did demonstrate there was a very, very large effect on the survival of bark beetles. And now with this new, using a different material, I'm going to get a much better number, except that I don't have very many trees. So it's the first demonstration of successful application of predator exclusion to mountain pine beetle. It's going to be a really good tool that can answer a lot of questions about mountain pine beetle. And then the emergence traps, well, I had these emergence traps on a number of trees, and this is one of the problems of the study for me was by the time I started the study 3 years ago, the bark beetle epidemic was over. There were only a few trees here and there that were infested with beetles. I couldn't find large enough numbers of trees to get a really precise measurement on anything with the bark beetles. I had 3 experimental trees in 2015, last year, which is the ones I'm now rearing beetles out of. I only found 2 trees that were suitable for setting up the experiment. So while I did demonstrate a really good, successful way of measuring this, we still have to get this out in the field where there is an epidemic going on, and we can really get enough experimental trees to get some good numbers. From the emergence traps, there are lots of insects which came out of the trees that are known to be associated with other pine trees. Pine trees I was working on were limber pine. Those are the only trees within like 300 miles of Laramie that still have any bark beetles in them. Ponderosa pine, unless I want to go up to Black Hills, there's practically no activity. Lodgepole pine, practically no activity. It's been that way for several years. So the only thing I could find that was nearby was the limber pine, which in a way that's kind of good because nobody's really studied this system in limber pine. There's been very little study. Lots of study has gone to lodgepole pine, ponderosa pine, important timber trees, and very little study to lodgepole pine or to limber pine. But the insect community in limber pine looks very much like it does in lodgepole and ponderosa pine. So many of these associates that were known to be with mountain pine beetle and those other pine trees are also present in limber pine. But here's the really cool part to me. Lots of associated insects, so the community is not identical. There are other insects in limber pine that are not found in the lodgepole or ponderosa pine. This is a very cool little wasp. It's pretty tiny. They're about 2 millimeters long. The year that I set up my first predator exclusion experiment, the following year, we found swarms of these little guys. I mean, they were super numerous. And it turns out that they don't attack mountain pine beetle, they attack a secondary beetle. So there are lots of other beetles, lots of other insects that wait to the mountain pine beetle to disarm the tree. The tree no longer has defenses, and all these other insects take advantage of that resource. That's what this wasp attacks, one of those other beetles. Another really cool one is a new predator. So there's some predaceous insects, and this belongs to the order Hemiptera, the true bugs. And these are known in other bark beetle systems. They are known to be present with southern pine beetle. They're known to be present with many of the different pine engraving beetles. And they've been demonstrated to be predators on the associated bark beetles. They predate on the larvae and the eggs of other bark beetles. And this is a pretty tiny insect, also about 3 millimeters. This insect is most likely a predator on pine beetle eggs and larvae. We haven't proven that yet, but just given the relationship related insects with their bark beetles, we think that's what this is. And this is an important predator. It came out of the trees in very large numbers. So we'll study this further and see what we get from this. So when I get those samples, the jars of insects out of my emergence traps, that is the coolest thing to me. That's like I look at one of those and I feel like I'm opening a Christmas present. I get all these new things. So just to give you an idea how I feel about this. So some final thoughts here. These methods don't just work with mountain pine beetle. They'll work with any insects that live under the bark of trees or inside of trees, standing trees. So this is going to have wide application. And it's going to be, we're going to be able to answer some really, really tough questions that we haven't been able to we didn't really have good methods of looking at before. And I'll give you an example of one of those. Most of you probably know that the mountain pine beetle has been expanding its range due to warming. So areas in the northern Canadian Rockies and the eastern Canadian Rockies that were not climatically suitable in the past for these beetles to reproduce in epidemic proportions are now supporting mountain pine beetle epidemics. And these mountain pine beetle epidemics are much more severe than the ones that occurred in its classical range. As bad as it looks here, they were much, much worse in Canada, in these areas. And one of the reasons that happened, I believe, and it's been brought up in research papers in the past, but nobody's actually done a study, is that the natural enemies of bark beetles in those areas are not adapted to mountain pine beetle. This type of experiment, the predator exclusion experiment, can be used to test that hypothesis. So a couple more really cool insects. This is another wasp we found associated with mountain pine beetle and limber pine. It's not described, it's an undescribed species, it's a new species. This one I put up here just because it's so pretty, and it isn't, but it's a known associate. And then, thanks to lots of people helped me out. I was funded by a competitive grant from the Agricultural Experimental Station, so thank you guys. And if you have any questions, I'm ready to sing. Yeah.
So you say the epidemic is about over up here,
Yeah.
on the downhill side?
It's beyond the downhill side. It is over.
Really?
Yeah.
Well, I just got back from Laramie Peak and I didn't notice any pine beetle kill up there.
Yeah,
Is it different trees or what's the deal?
No, this is really interesting, and I don't think we really know why. There are a number of areas where there are forests that seem perfectly suited for bark beetle reproduction. They didn't have serious outbreaks. So the bighorns are that way also. Yeah.
Did drought conditions play any role in the population of the pine beetles?
Absolutely, absolutely. Under drought conditions, the trees can't produce as much of the defensive chemicals that they produce in response to attack from insects, and they can't defend themselves as effectively.
Do you think you showed a dramatic decrease in the beetle when you had the predators?
Yes.
Do you think that the predators would be overwhelmed during the epidemic, or would they also increase in the population?
That's a real good question, and they are overwhelmed. The bark beetles goes into a new shifts. It's a new life cycle regime shift. They now have so much more food that their populations just grow so rapidly that the predators can't keep up with them. So later on in an epidemic, you start to see the numbers of predators catching up. And it is believed, at least in some bark beetle systems, maybe not with mountain pine beetle, that the predators, when they do catch up, are actually the agent responsible for causing the population collapses. And it may be true for mountain pine beetle, except that mountain pine beetle generally tends to go through an area so fast and pretty much work it over so completely that by the time that area has no more suitable trees for bark beetles to reproduce in, the predators still haven't caught up.
I found out interesting thing a couple of years ago. A friend of mine from Whitehall, Montana, brought some logs down from an old stage stop, and I milled them. I have a sawmill. And when I got to milling them, there was blue stain in those logs from the very early 1900s.
Yeah, oh yeah, yeah. So mountain pine beetle is a native insect. The epidemics have been happening for millennia. They evolved with these trees. There's evidence of epidemics going back thousands of years, and part of that evidence is from lake sediments. So they can go into lake sediments, and there are certain parts of the beetles that don't decay very easily. Like the elytra, beetles have some very hardened parts of their bodies that don't decay very easily. And you can see layers of sediment where there's huge numbers of remains of beetles and lots of other sediment layers where there's none, or minimal. And you can also see that in glaciers.
I'm figuring about etymology. You got like, in microbiology, we go with like logarithm reduction, and you got maybe almost a 2-log reduction. Is that significant? Where we would say we need a 5-log reduction in food safety, for instance?
I'm not exactly sure. You mean
Well,
like 2 orders of
I
magnitude
understand.
reduction in population?
Yeah,
That's pretty significant.
okay.
So the kind of things we saw, say for whatever reason, the densities of beetles during flight times was much higher in these areas in Canada, in the new regions where the mountain pine beetle hadn't been in epidemics before, at least at historic times. The beetles attacked a lot of unsuitable host trees. When mountain pine beetles in very high numbers, they attack trees that can't reproduce in very well. That includes smaller trees, which don't have very thick phloem, not a good food source for them. So they kill lots of younger trees. They have a much higher mortality of the larger sized grass trees. And that's just, and I can't really, I don't really know if we have a good way of measuring population densities, but it did a lot of things that due to that increase in population density. And part of that is thought to be because the predators are not well enough adapted there, or not particularly adapted to mountain pine beetle. And they'll adapt real quick, those predators, because insects have really short life cycles. So that's going to happen a lot faster than trees developing resistance